Hi everyone. In this lecture, I'm going to talk about different cell types. So everything alive in the world is made up of cells, and there are only two types of cells in the world. So if you're alive, you're either made up of prokaryotic cells or eukaryotic cells. There's no other cell type. And this is what we will focus on in the next two lectures. We're going to talk about what prokaryotic cells are, what eukaryotic cells are. And this lecture specifically is going to focus on prokaryotic cells. And then in another lecture video I make, we'll talk about eukaryotic cells. But just to give a background, prokaryotic cells, organisms that are made up of prokaryotic cells, are archaea and bacteria. So all bacterial infections, any bacteria that you see in the ocean and the environment, when you look at that bacterium, it's made up of a prokaryotic cell. And then archaea organisms, which we will talk about more in class, are also made up of prokaryotic cells. Everything else alive in the world is made up of eukaryotic cells. So when we look at humans or animals, ourselves, our cells are eukaryotic cells, and they're just different cell types, and we're going to focus on the similarities and the differences between these. In our lecture outline, what I would like to talk about in this lecture today is just give a really quick overview of the differences between prokaryotic cells and eukaryotic cells, and then we will focus on prokaryotic cell structure. So the reason why it's important to learn cell structure, students ask me a lot of the times, like, is this just memorization? And it's not just memorization. When we really, as scientists, understand cell structure, we can create drugs to target them. So for example, today we're focusing on prokaryotic cell structures, and bacteria are made up of prokaryotic cells. So bacteria cause a lot of infections, and if you're creating an antibiotic or a drug to target bacteria, it's good to understand their cell structure so that you can create a drug to target those cell structures. But we also need to know eukaryotic cell structures because we're eukaryotic organisms, and so you don't want to create a drug that harms something in prokaryotic cells that's also present in eukaryotic cells, because you also end up harming the host. So that's what we're going to talk about today. And then with prokaryotic cell structures, we're going to talk about all the things in a prokaryotic cell that you find, so all the different components, and then the function of each component. Okay, to start off again, to just recap, anything alive, which we call an organism, is made up of cells, and cells are either prokaryotic or eukaryotic cells. The main difference between these two cell types are that prokaryotic cells do not have a nucleus and eukaryotic cells have a nucleus, and I'll talk about what a nucleus is. So if I were to draw these cell structures in lab, I would draw a circle for each cell type, and then when we look at eukaryotic cells, which we tend to call the more complex cells, and we call prokaryotic cells the simpler cells, eukaryotic cells inside the cells have a membrane-bound structure that we call a nucleus, and the function of a nucleus is to house the DNA or to hold the DNA. So both prokaryotic cells and eukaryotic cells, all cells in the world have DNA. Eukaryotes have a nucleus, and the function of the nucleus is to protect the DNA. A common question is, well, what about prokaryotic cells? Don't they need to protect their DNA? The answer is yes, but the DNA is much smaller and much simpler, and prokaryotes also do some fancy things. So having a nucleus would also get in the way of getting more DNA, which we will learn about when we talk about bacteria. So that's the main difference between prokaryotic cells and eukaryotic cells. And by the way, prokaryotic, it comes from the word pre-nucleus, so karyote is nucleus. So prokaryotes came before eukaryotes, they don't have a nucleus, and eukaryotes have a nucleus that protects their DNA. So if you forget everything from biology, just remember in the future, if anyone ever asks you what's the main difference between prokaryotic and eukaryotic cells, prokaryotic cells have no nucleus and eukaryotic cells have a nucleus, and its function is to house the DNA. So now we're going to talk about the similarities between all cells. So what are things that are similar between prokaryotic cells and eukaryotic cells when we look at them? Again, organisms that are made up of prokaryotic cells, or we call prokaryotes, are bacteria and archaea. All other organisms in the world, Plants, protists, amoeba, humans are made up of eukaryotic cells, or we call them eukaryotes. The similarities between all cell types, or both of these cell types, are that all cells have a plasma membrane. Let me go back. Okay, a plasma membrane. So a plasma membrane and a cell membrane mean the same thing. Sometimes you'll see people use both words. It means the same thing. When someone says cell membrane, plasma membrane, it's just the outside lining of the cell. So all cells in the world have a plasma membrane, and the function of the plasma membrane is to protect what's inside the cell. So this is the plasma membrane, and this is a eukaryotic cell plasma membrane, just the outside. And then another thing that we see in all cells is all cells in the world have ribosomes. We will talk about what these are, but ribosomes in these little cartoon images are these little dots here. The function of ribosomes inside cells is to make protein. Protein is very important for cells. We'll learn about it later. And then finally, all cells in the world have DNA. DNA is a genetic material. So if I were to ask you on an exam, what do we see in all cells? What are the similarities between prokaryotic cells and eukaryotic cells? I want everyone to tell me that all cells have a plasma membrane, all cells have ribosomes, and all cells have DNA. So now we're going to talk about the differences between the cells. So when we look at prokaryotic cells, bacteria and archaea are made up of prokaryotic cells. They do not have a nucleus, unlike eukaryotes, which have a nucleus. They have one circular piece of DNA. So we call DNA and chromosome mean the same thing. A chromosome is DNA, and they just have one, and it's circular. So in prokaryotes, the DNA is circular. The DNA is concentrated in a region called the nucleoid. Nucleoid and nucleus are very different things. Remember, prokaryotes do not have a nucleus, but they have a nucleoid. Nucleoid just means the region where we see the DNA. So the DNA bundles up, and that region where the DNA is called the nucleoid. There are no organelles or histones in prokaryotes. We'll learn about what that is, but prokaryotes do not have that. And when we look at bacteria, which are made up of prokaryotic cells, they also have a cell wall made up of peptidoglycan. So remember, all cells have a plasma membrane. Bacteria also have a cell wall, which we will learn about, and it's made up of peptidoglycan. And archaea have a cell wall, and it's made up of pseudomurine. And then when we look at prokaryotic cells, they divide by binary fission, which you will learn about, meaning that when they go to make a copy of themselves, they just basically split. That's what binary fission is. So there is no sex involved in prokaryotes. That's why bacteria, like if you have a bacterial infection, it can quickly go from being very mild to very strong. A strong bacterial infection means you have a lot of bacteria, because bacteria can quickly divide and make copies of themselves. And that process is called binary fission. So these are the differences. So again, prokaryotes do not have a nucleus. They have a circular chromosome. The DNA is in the nucleoid, and they don't have organelles. Now let's talk about eukaryotic cells. So all other organisms are eukaryotic cells. If you're not bacteria and you're not archaea, you're a eukaryotic cell. So when we look at eukaryotes, they do have a nucleus, and the purpose of a nucleus is to hold the DNA. They have paired DNA or paired chromosomes in the nucleus. So in the nucleus, when we look at the DNA, it's paired, unlike the one circular DNA that's just floating around in prokaryotes. They have eukaryotes have organelles and histones, which we will talk about. And if they do have a cell wall, such as in plants, it's made up of polysaccharide, not peptidoglycan, like we see in bacteria, which are made up of prokaryotic cells. And we're going to talk about all of these things in details. This is just a big overview summary of the differences. And then when we look at eukaryotic cells, they divide by mitosis. So if any of you have taken the general biology class, you'll learn all about mitosis. But because this is microbiology, we're going to focus on prokaryotes and bacteria mainly. Okay, I want to just stress this before we move on. So I always like to ask students, are viruses prokaryotes or eukaryotes? And the answer is they are neither, because viruses are not alive, therefore they're not made up of cells. So if you're ever asked on a test, are viruses prokaryotic cells or eukaryotic cells, they're neither. Viruses are not alive. They can only divide when they take advantage of a host, and we'll learn about it. So they're made up of neither, not prokaryotes, not eukaryotes. So if you're asked coronavirus, is it made up of eukaryotic cells or of prokaryotic cells? I want everyone to tell me neither. But if I asked you the bacterium E. coli or the bacteria Salmonella that causes food poisoning, what type of cell is it made up of? You're going to say prokaryotic cells. Okay, now let's talk about human cells. So this is a trick question I always ask students. What type of cells are humans made up of? And I just spent this whole time telling you that humans are made up of eukaryotic cells. And I want everyone to know that yes, our own cells are eukaryotic cells, like my own skin cells, but we have so much bacteria in us that we will learn about. So when you look at the composition of a human, inside a human, you have both prokaryotic cells and eukaryotic cells, but human cells themselves are eukaryotic cells. So we have so many microbial cells within us. So just keep that in mind, that bacteria play a big role within us. Okay, now the rest of this lecture is going to focus on prokaryotic cell structure. So we're gonna dive in detail about what prokaryotic cells look like and the structure and function of everything I talk about. So we're going to talk about the plasma membrane in prokaryotes, what its structure is and what its function is. Why do cells have a plasma membrane? Remember, a plasma membrane is found in all cells, prokaryotes and eukaryotes. We're going to talk about the cytoplasm. So in cells, there is the inside component of cells inside the plasma membrane, that's called the cytoplasm. We're going to talk about the nucleoid. The nucleoid is just where the DNA is concentrated. It's not the nucleus. And then we're going to talk about ribosomes. So inside prokaryotic cells, we see ribosomes. And just know a lot of times I'll tell you inside bacteria, I'm also talking about prokaryotic cells. So we see these structures called ribosomes, and their function is to make proteins, which basically they do everything in our cells and in prokaryotic cells. And then we're going to talk about the cell wall. So the cell wall may be found in some prokaryotes and is not present in all prokaryotic cells. That's why I have plus or minus. So these things are found in all prokaryotic cells. Some prokaryotic cells, in addition, have a cell wall, some don't. Some prokaryotic cells have a capsule, some don't. We will learn about what that is. Some have flagella, some don't. Flagella is used for motility. Some have pili. Some make endospores. We're going to learn about what that is. Okay, so we're going to go through all of these. I'm going to start off with the plasma membrane. The plasma membrane is the same thing as a cell membrane, is the same thing as cytoplasmic membrane. So you'll see in textbooks, or if you're talking to someone, they use all these terms. It all means the same thing. When we look at the plasma membrane, the structure, what it's made up of, is it's a phospholipid bilayer, meaning that there are two layers. It's a phospholipid, and so there is fat, that's what lipid means, and there are phosphate groups. So when we look at the cell membrane, we see two layers. The head is polar and it's hydrophilic, and the tails are nonpolar and they're hydrophobic. So when we look at this cell membrane, it's easy for hydrophobic components to move across the cell membrane because the inside of it is mainly hydrophobic. And then there are also proteins all throughout the cell membrane or plasma membrane. So when we look at the cell membrane, the whole thing is a phospholipid bilayer, and throughout it there are proteins. Okay, the function of the plasma membrane. The plasma membrane, the main function, the reason why cells have a plasma membrane is to protect the cell components, to protect what's inside the cell. That's mainly it. Remember, inside the cell there's DNA. DNA is probably the most important component in all cells, whether you're a prokaryotic cell or a eukaryotic cell. So its function is for protection. It's also semi-permeable membrane. Semi-permeable is just a fancy word meaning that it lets some things permeate or diffuse across and other things not. So it's half permeable. And we'll talk about transport. And it's also important for transport. For cells, you need for things to get in and out. For example, you need nutrients to get into cells, and you need toxic material and waste to get out of cells. So the plasma membrane is important for transport across the cell. And then it's important to sense the environment. So bacteria use their plasma membrane to sense whether they should go towards an environment or get away from like your immune cells, which are trying to kill it. And then also the plasma membrane is important for making energy for cells or making ATP. In cells, the energy molecule is called ATP, and you'll learn about it later. And so here's a good video that describes the cell membrane.
The cytoplasmic membrane of a prokaryotic cell is the main gateway for movement of molecules in and out of the cell. This membrane may also be called the cell membrane or plasma membrane. Cytoplasmic membranes in bacteria are comprised of a phospholipid bilayer. Each phospholipid contains a phosphate head located on the outer part of the bilayer, attached to a glycerol and two fatty acid, which face the core of the membrane. The phosphate is negatively charged and hydrophilic, while the tail portion of the phospholipids is hydrophobic. Thus, cytoplasmic membranes are very effective at blocking passage of many molecules, while other molecules are allowed to pass.
So this is what we mean when I say that the cell membrane is semi-permeable. It means that it lets some things out and some things in, and other things it doesn't let them move across. A lot of times students think that movement is just in cells, but we also need to let things out of cells. So that's what semi-permeable, and the reason why it's semi-permeable is because when we look at the structure, we have the phospholipid bilayer, which inside is hydrophobic and outside is hydrophilic.
Cytoplasmic membranes also contain two types of proteins. Integral proteins often span the entire bilayer and act as channels for transport of molecules that normally wouldn't be able to penetrate the bilayer. Peripheral proteins are loosely attached to the membrane on one side or the other. Some of these may be glycoproteins.
Okay, I'm going to stop it here. So the main take-home message I want you to take is when we look at the structure of a cell membrane, it's made up of a phospholipid bilayer, that is all of this. And within this phospholipid bilayer, within the cell membrane, we see a lot of proteins, and these proteins are important for transport. So that's the structure. The structure of the cell membrane is a phospholipid bilayer with proteins. I want everyone to take that as a take-home message. Okay, now one of the functions I said of the plasma membrane is transport. So we're going to talk about how materials move across the cell membrane, how they go in and out of a cell. And there are two main types of transport that we're going to focus on. There's passive transport and active transport. The main difference is that passive transport requires no energy. So molecules move in and out of the cell and you don't need any energy. And then with active transport, it is active, so it requires energy, and the energy molecule of cells is ATP. That's the main difference. So there's two types of transport across the cell membrane. There's passive transport and active transport. And then within these two main groups, we're going to talk about it a little bit more in detail. Okay, so passive transport do not require energy, meaning that, like for example, water gets in and out of cells without requiring energy, so it's a form of passive transport. And with passive transport, molecules always move from where there's a lot of them to where there's less. So for passive transport, you go from high to low concentration. So water will move to where there is less of it, always. Again, for passive transport, it's no energy and you're going from high to low concentration, meaning you're going with the concentration gradient. With active transport across cell membranes, energy is required and the energy molecule is ATP. So to move things across, you think about moving big molecules. That's an example of active transport. Energy is required, and this is when molecules go from where there's just very little of them to where there's a lot. So from low concentration to high concentration. And we say that this is going against the concentration gradient. So anytime you hear someone say you're in active transport, you're going against the concentration gradient, it means you're going from low to high concentration. That requires energy. Okay, now we're just going to focus on passive transport. Again, passive transport requires no energy and molecules are always moving from high to low concentration. So the three types of passive transport across cell membranes are simple diffusion, facilitated diffusion, and osmosis, and we're going to talk about each one of these. So simple diffusion is when molecules move from an area of high concentration to an area of low concentration, and they're small. So anytime you have a small molecule and it goes from high to low concentration, we say that that's just diffusion. Facilitated diffusion, facilitate means to help. So facilitated diffusion is when you have big molecules that need to move across the cell membrane, so you need a protein. So facilitated diffusion means that you require a protein to move the molecule from high to low concentration. And osmosis is just the transport of water. So osmosis is just when water gets through the membrane, whether it's in or out. That's a type of passive transport, and we specifically call that osmosis. So again, 3 types of passive transport. All of them require no energy. All of them are molecules going from high to low concentration. We have diffusion, facilitated diffusion, and osmosis. Diffusion or simple diffusion is just when small molecules move across the cell membrane from high to low concentration. Facilitated diffusion is when you have big molecules trying to move across, so you need a membrane protein. So that's why it's called facilitated. And then osmosis is just the movement of water. So it's just the transport of water. And by the way, a lot of times students ask me, osmosis, is water just going across the cell membrane, or is there a protein that helps water get through? And the answer is there's both. So water is such a big part of cells that water can get diffused in and out of cells, or it can also go through facilitated diffusion, meaning that there's protein in the cell. There's proteins in the cell membranes that let water in or out. So here's a good video that explains all 3.
Passive transport includes diffusion, non-specific and specific facilitated diffusion, and osmosis. All of these processes occur along electrochemical gradients and do not require the expenditure of energy in the form of ATP. Diffusion is the net movement of a chemical down its concentration gradient, that is, from an area of high concentration to an area of lower concentration. This diffusion requires no energy expenditure by the cell, as the concentration gradient drives the diffusion.
So diffusion is just small molecules going in and out of the cell membrane. There's no energy required, and you're going from high to low concentration. So oxygen will go from where there's a lot of it to where there's a little of it, and there's no protein required.
Small uncharged molecules, such as dissolved oxygen, dissolved carbon dioxide, small alcohols, and small fatty acids, can freely diffuse through the cytoplasmic membrane. Once equilibrium is reached, molecules will still move across the membrane, but there is no net movement in a particular direction.
Okay, so this was diffusion. The next thing we will talk about is facilitated diffusion. Facilitated diffusion is just when you have a protein that you're using in the cell membrane, and then osmosis is the movement of water. Okay, so we talked about, in the last slide, we talked about diffusion and facilitated diffusion. Again, the difference is with regular diffusion or simple diffusion, you don't need a membrane protein. With facilitated diffusion, you need a membrane protein. And now the last type of passive transport I'm going to talk about is osmosis. Osmosis is just the movement of water across a cell membrane, where water goes from high concentration to low concentration. And remember, this requires no energy, so it's not a form of active transport. It's still passive transport. And water can move through the cell membrane, either just through simple diffusion or through facilitated diffusion using aquaporins. Aquaporins are just cell membrane proteins that let water through. So again, the 3, I think I have some summarize it. Okay, before I get more into this, I know I'm repeating this a lot, but I want this to be a take-home message because they do ask it on nursing tests for a lot of you. 3 types of passive transport are simple diffusion, facilitated diffusion, and osmosis, and right now we're focusing on osmosis. So there's something in cells called osmotic pressure, and it talks about the pressure that's put on cells by the water concentration in the solution they are in, so in their environment. So I want everyone to know that water will always move where there's more solutes. Solutes are like salts or sugars from chemistry. I know chemistry is a prerequisite, so hopefully you guys remember that, but if you don't, solutes are just, like for example, salts. Water will always move where there's more solutes. So if you have a cell and there's more, let's use salt as an example of a solute, there's more salt inside the cell than outside the cell, water will move toward inside the cell. If you have a cell and there's more salt outside the cell than inside the cell, water will move outside the cell. Water will always go where there's more solutes. So everyone just keep that in mind. If we're drawing a cell, water will always move where there's more salts or sugars. So if there's more salt inside the cell than outside the cell, water will move in the cell. If there's more salt outside the cell than inside the cell, water leaves the cell. So water will go outside the cell. It will always go to where there's more solutes or salts or sugars. Now, the types of solution we can put cells in. So if I was actually growing cells in lab, or just cells in the environment, but let's think of cells in the lab because it's easier. There are 3 types of solution that you can put cells in. You can put cells in an isotonic solution. Isotonic means that the salt concentration in the solution is the same as the salt concentration inside the cell. You can put cells in a hypotonic solution. Hypo means less, so the solution has less salts than the cell. And you can put cells in a hypertonic solution. Hyper means more, so a hypertonic solution has more salts or solutes than the cell. So again, the 3 types of solution are isotonic solution, hypotonic solution, and hypertonic solution. In an isotonic solution, that solution has equal amount of solutes as the cell. A hypotonic solution, hypo means less, has less solutes than the cell. A hypertonic solution has more solutes than the cell. It's hypertonic. So when we take a cell and we put it in an isotonic solution, so imagine if I had a flask and I put cells in it, like bacterial cells. Isotonic means that my solution has an equal concentration of solutes as the cell. So my question to you is, where does water move? Remember, water always moves where there's more solutes, but if there's equal solute concentration in the solution and there is equal concentration in the cell, water will move equally in and out of the cell. A lot of times students will say, oh, nothing is happening. No, something is happening, it's just at an equal rate. If there was nothing happening, the cell would die. So now we're gonna focus on hypotonic and hypertonic. Everyone remember that water will always move where there's more salts. Okay, so now let's talk about a hypotonic solution. Remember I said a hypotonic solution is a solution that has less salts than the cell itself. So if you have a solution and there's less salts in it than the cell has more salts. Remember I said water will always move where there's more salts, and I ask you on a test, how does water move in a hypotonic solution? You're gonna think about it and you're gonna say, okay, a hypotonic solution has less salts than the cell, so water will move where there's more solutes, so it will go inside the cell. So water will enter the cell and water will move into the cell, so the cell will keep getting bigger and bigger and bigger and bigger and bigger and burst eventually. If we put a cell in a hypertonic solution means that there's more salts in the solution than the cell. So that means that the water in the cell will leave it because it wants to go to where there's more solutes. And when water leaves the cell, the water will keep leaving it and it'll shrivel and shrink. So that's what happens. So the reason why this is important, the whole types of solutions, is if you're a scientist and you're growing cells in lab, if you are growing cells in a hypotonic solution, the cells will grow so big and burst and eventually die. If you grow cells in a hypertonic solution, if your solution is so salty, the water in the cells will leave and the cells will shrivel and die. So it's always important in general, depending on what you're trying to do, to figure out the salt concentration of your cells so that you can grow them in an isotonic solution. That's one reason. So if I'm trying to grow or culture bacteria in lab, culture bacteria means grow them, I want to make sure that I grow those cells in an isotonic solution, because a hypotonic solution is bad and so is a hypertonic solution. Okay, so we finished with the types of transport and passive transport. So to summarize, the three types of passive transport are diffusion, or sometimes just called simple diffusion, facilitated diffusion, which requires a protein, and osmosis, which is the movement of water. So if I ask you on a test, what are the three types of passive transport, I want everyone to tell me diffusion, facilitated diffusion, and osmosis, and know the difference between the three. And remember, in all passive transport, no energy is required, and also molecules are going from high to low concentration. And then I want you to know the different types of solutions and what happens to cells in these different types of solutions. Now we will talk about active transport. Active transport across a cell membrane is when molecules move across a cell membrane, but they require energy. So with active transport, you require energy. For cells, that means that you're using a molecule called adenosine triphosphate, or ATP for short. You require a protein for all active transport, and molecules are going from low to high concentration, which means that they're going against the gradient. So they're going from low concentration to areas of high concentration. And whenever you're going from low concentration to high concentration, this requires a lot of energy. So that's what active transport requires. It's three things. It requires energy, it requires a protein in the cell membrane, and molecules are going against their gradient, meaning they're going from low to high concentration. And okay, so it looks like I summarized transport here. This was the end of transport. Remember, the two big classes of transport in cells are molecules will either move across membranes through passive transport or active transport. With passive transport, we have three more categories. We have simple diffusion, facilitated diffusion, and osmosis. And then we have active transport, which is what I just described. And this is all involving the plasma membrane. So now we're going to move on to the prokaryotic cell wall. So if I were drawing a cell on the board in class, we have the cell membrane, or the plasma membrane, that we just talked about. Some cells, some prokaryotic cells also have a cell wall. A cell wall is outside the cell membrane. So when you think about it, it's probably for more protection, and that's exactly the function of prokaryotic cell walls. So prokaryotic cells have a cell wall outside the cell, or cytoplasmic or plasma membrane, and the function of the cell wall is to prevent osmotic lysis, because we were just talking about osmotic pressure. It's for protection. That's why the cell wall is present. So if you are a bacterium and you have a cell wall, that means that you probably have more protection than a bacteria that doesn't have a cell wall. So again, a cell wall is always outside the cell membrane, and it protects the cell membrane, and it also contributes to pathogenicity. Pathogenicity is just a fancy word meaning that the microbe can cause more disease. So microbes that have, bacteria that have a cell wall can be pathogenic, meaning can cause disease, because a lot of times your immune system may not be able to kill the cell as effectively because it has that layer. We're going to learn more about it in detail, but that's the function of the cell wall. It's to protect the cell membrane and to prevent osmotic lysis. When we look at the bacterial cell wall, the cell wall in bacteria is made up of a component called peptidoglycan. So again, in bacteria, if I ever ask you what are cell walls in bacteria made up of, they're made up of peptidoglycan. And when we look at peptidoglycan, it's made up of NAG and NAM molecules. NAG stands for N-acetylglucosamine, and NAM stands for N-acetylmuramic acid. They're just molecules. So outside the cell membrane, there's a cell wall, and it's made up of peptidoglycan, and peptidoglycan is made up of NAG and NAM molecules. The reason I want you to know that it's made up of NAG and NAM molecules is because later on we're going to learn about antibiotics, and I'm going to tell you that there's an antibiotic that stops NAG and NAM molecules from coming together, just so you keep all of it together in your mind when we talk about this later. So in bacteria, we can classify them, bacteria that have a cell wall, as gram-positive or gram-negative bacteria. And I'm going to talk about what that is. So when sometimes people will say, okay, there's gram-positive bacteria and there's gram-negative bacteria, and they have differences in their cell wall. That's the main difference. So when we look at gram-negative bacteria and gram-positive bacteria, for both of them, their cell wall is made up of peptidoglycan. Remember, bacteria, the cell wall is made up of peptidoglycan, but there are some differences. Gram-negative bacteria have thin peptidoglycan, and they also have an outer membrane. So they have their cell membrane that all cells have. They have one layer of peptidoglycan, and then they have an outer membrane. That's what gram-negative bacteria look like if you think about it. Here in this cartoon, they're just taking a chunk, but remember, it's a circle. And then when we look at gram-positive bacteria, they have thick peptidoglycan, so they have many layers of peptidoglycan. That's the main difference. So if I ask you between gram-negative bacteria and gram-positive bacteria, which one has an outer membrane? Gram-negative bacteria has an outer membrane. Which one has thick peptidoglycan layer? It's gram-positive bacteria. So in this cartoon, the peptidoglycan is purple. And this is going to become important later on when we talk about infection. But these are the two main classes of bacteria that have cell walls. They're gram-positive or gram-negative. Gram-negative bacteria have an outer membrane and thin peptidoglycan. Gram-positive bacteria have thick peptidoglycan, a lot of layers of peptidoglycan. So let's just focus on gram-positive bacteria. So examples of gram-positive bacteria, which I do not want you to memorize, but just to give you an idea of bacteria that are what we call gram-positive, are Bacillus, Staphylococcus, Streptococcus, many, many others. So Staphylococcus, if any of you guys know this, like Staphylococcus epidermis is a bacteria that we have on our skin. Streptococcus bacteria, one species of it causes strep throat, the really bad sore throat. These are what we classify as gram-positive bacteria. So if someone has strep throat and I take a little bit of their throat sample and I look at it under the microscope and do a few things which we will talk about, Streptococcus bacteria are gram-positive, meaning they have a thick cell wall, they have lots of peptidoglycan. So they have thick peptidoglycan. They also have teichoic acids, and teichoic acids help link all the peptidoglycan layers together because there's so many, and they stabilize them to the plasma membrane. So again, with gram-positive bacteria, they have a lot of peptidoglycan. Now, I want everyone to memorize this. We're going to talk about it later. There's gram-positive and gram-negative. The mnemonic I used to use to memorize that as a student is positive peptidoglycan purple. So in lab, when we stain gram-positive bacteria, they always stain purple. So I always like to think of it as positive, purple, lots of peptidoglycan. So PPP. That's how you're going to memorize it. Gram-negative bacteria, on the other hand, examples of it are E. coli, Pseudomonas, Salmonella bacteria, many other bacterial species. They have thin peptidoglycan, and they also have an outer membrane. The outer membrane is made up of polysaccharides, which are sugars, lipoproteins, fatty proteins, and phospholipids. So we'll talk about these molecules later on. And so the outer membrane is a bilayer, and this bilayer helps protect the bacteria even more. It's really good for the bacteria. It's kind of bad for us because your immune system has a hard time targeting gram-negative bacteria. And again, the lipopolysaccharide, the LPS, forms part of this outer membrane. So there's something called lipopolysaccharides with our fatty sugars, and these make the bacteria more pathogenic or cause more disease. So their lipid A, that's part of LPS, is a toxin, and we're going to talk about toxins later on. It's an endotoxin, and these are the toxins that make you sick. So the summary of this slide is that gram-negative bacteria have thin peptidoglycan, and they also have an outer membrane. The outer membrane is made up of polysaccharides, lipoproteins, and phospholipids, and components that can make you sick, such as lipid A, which is an endotoxin. So the way that we differentiate gram-positive from gram-negative bacteria in lab, and the reason why this is important is if someone comes to you and they have an infection, whether it's like a kidney infection, a stomach infection, a throat infection, Knowing what type of bacteria it is, gram-positive or gram-negative, is important because some antibiotics we prescribe for gram-positive bacteria, some we prescribe for gram-negative bacteria. So it's important for treatment and also just for having a better understanding for what the infection is. So in lab, there's something we do called a gram stain, and I have a lab video for you all to learn later. And the gram stain can differentiate gram-positive bacteria from gram-negative bacteria. When we look at these types of cells, this stain, the order of it, you do a lot of staining procedures in the gram stain, and in the end, gram-positive bacteria always stain purple and gram-negative bacteria stain red. That's why I told you if you see purple bacteria under the microscope, you will tell the doctor that this person has a gram-positive bacterial infection. So the way the gram stain is done is you take, like for example, the easiest thing I can think of is if someone has like a sore throat and you want to figure out, thinking that it's bacterial, not viral, you take a sample, you put it on a slide, and you do a gram stain. The way you do a gram stain in lab is once you have your bacteria on a slide, you put a stain called crystal violet. So crystal violet is violet, that's the color of it. It turns the cells purple, all cells, whether you're gram positive or gram negative, they turn purple. Then what you do after you add the violet stain is you add something called a mordant, and the mordant is iodine. It's just a liquid. You squeeze a little of iodine on it. Iodine helps the primary stain, the violet, stick more to the cells. So now your cells are still purple, all your cells. And then you add alcohol. Alcohol dehydrates peptidoglycan. So if you do not have a lot, it ends up causing you to lose the purple color, and I'll talk about it. And then you add a secondary stain, which is red. So for gram-positive bacteria, the crystal violet sticks to the cells, and it sticks really strong because gram-positive bacteria have a lot of peptidoglycan, so they stain purple. For gram-negative bacteria, when you add the alcohol, the crystal violet washes out and the cells become clear, so that's why we add a counter stain, which is red. It's called safranin, and we talked about it briefly in another lecture. So the gram-negative bacteria stain red. So this is why gram-positive bacteria stain purple and gram-negative bacteria stain red. So these were talking about gram-positive and gram-negative bacteria. The take-home message I want everyone to know is gram-positive bacteria have thick peptidoglycan and they stain purple. Gram-negative bacteria have thin peptidoglycan, an outer membrane, and they stain red. So these are the main differences between the two. Now, Mycoplasma bacteria are different. They're not gram-positive bacteria or gram-negative bacteria. They're unusual bacteria because they do not have a cell wall. So there is no peptidoglycan in Mycoplasma bacteria. Instead of having a cell wall, remember the function of a cell wall is protection. They have these molecules called sterols in their plasma membrane, and these make the plasma membrane strong to protect the cell components even more. So an example of Mycoplasma bacteria is Mycoplasma pneumoniae bacteria. This bacteria causes a disease or an infection called walking pneumonia. It's a very mild case of pneumonia or a lung infection where someone just feels like they have a cold, but you're very contagious. So it's the leading cause of pneumonia in children, and it affects someone's lung. So if someone comes to you in a hospital with just like pneumonia symptoms, they may have Mycoplasma pneumoniae. So if you were to send this sample to lab and they did a gram stain, it would not work because these bacteria do not have a cell wall. Remember, with a gram stain, we either get purple for gram-positive bacteria or red for gram-negative bacteria. It won't work on Mycoplasma bacteria at all. Okay, I'm going to continue this recording. I apologize, I stopped it. So one of my questions with this slide was, would you do a gram stain with Mycoplasma bacteria? And the answer is no, because they do not have a cell wall. So the gram stain won't work. So we have our gram-positive bacteria, gram-negative bacteria, and Mycoplasma bacteria, which have no cell wall. And instead of having a cell wall, they have sterols in their plasma membrane. Another type of prokaryotic cell that we talked about earlier was Archaea. Archaea are not bacteria, they're completely different organisms. I want to just have one slide to talk about their cell wall. Their cell wall is, they're not bacteria, so it's not made up of peptidoglycan. They are Archaea, just to remind you from the very first lecture, are prokaryotic organisms that often live in extreme environments, but now new research is actually showing that they can be everywhere like bacteria. They do not have peptidoglycan, but instead they have either no cell wall, so wallless, cell wall-less organisms, cells, or if they do have a cell wall, it's made up of pseudomurine. So if I ask you if Archaea have a cell wall, what is it made up of? I want you to know that it's pseudomurine. And then for bacteria, the cell wall is made up of peptidoglycan, and Mycoplasma bacteria do not have a cell wall, remember? So we do not do a Gram stain with them. Okay, to move on, I want to talk about penicillin. So we're going to learn all about antibiotics later on in the semester, but penicillin is an antibiotic that targets the cell wall. And when we look at bacteria, so we talked about gram-positive bacteria, gram-negative bacteria, and Mycoplasma bacteria. If I tell you an antibiotic targets a cell wall and bacteria, Mycoplasma bacteria have a cell wall, you will not prescribe penicillin because it will not work on someone who has walking pneumonia. So now we're left with gram-positive and gram-negative bacteria. Both of them have a cell wall. And when I say that penicillin specifically targets peptidoglycan, and remember, out of gram-positive and gram-negative bacteria, the one that has thick peptidoglycan, so PPP, is gram-positive bacteria. So with penicillin, we prescribe for gram-positive bacterial infections. So penicillin targets peptidoglycan. Gram-positive bacterial infections are really susceptible to penicillin. Susceptible means it works on them, so they're affected by it. Gram-negative bacterial infections, penicillin doesn't work that much on them, because if you remember, gram-negative bacteria have an outer membrane and they only have a thin peptidoglycan layer, so the drug really cannot get in because of that outer membrane. So if someone has a gram-negative bacterial infection, we generally look at other antibiotics, not penicillin. And specifically what penicillin does is it stops the cross-linking of NAG and NAM molecules in peptidoglycan. So it stops growing and dividing cells. It affects gram-positive bacteria mainly because gram-negative bacteria have an outer membrane. And can penicillin treat walking pneumonia? The answer is no, because it targets peptidoglycan, and walking pneumonia is caused by Mycoplasma pneumonia, which do not have a cell wall, do not have peptidoglycan. Would it work on Archaea? No, the cell wall in Archaea is made up of pseudomurine molecules, not peptidoglycan. So that's how I want you to think on a test. And just keep in mind that we have lysozyme, and lysozyme is our natural penicillin. So lysozyme is an enzyme that's found in human secretions such as tears. And so your tears are very good antibiotic components to target gram-positive bacteria. And we'll learn more about this later on. Okay, another cell structure I want to talk about to move on from the cell wall is a bacterial capsule. A bacterial capsule is when we look at a cell, you have a cell membrane and then a cell wall, and then bacteria also sometimes have a capsule. So it's a third layer of protection, and it's external to the cell wall, meaning it's outside the cell wall. It's made up of sugars. Polysaccharides are sugars, and also sometimes proteins. Polypeptides are proteins. It's called a slime layer if it's not tightly organized, and it's not present in all bacteria. So some bacteria have capsules, some don't. As you can imagine, if you have an infection with a bacteria that does make capsules, it's harder to treat because it's so hard to have a drug or your immune system get through all the layers, get through the capsule, the cell membrane, the capsule, the cell wall, the cell membrane. So bacterial capsule is important for protection for bacteria. It also helps bacteria stick to each other and to surfaces. This is not good for you because when bacteria stick to each other, they're harder to treat with antibiotics, and sticking to surfaces are like bacteria sticking to your lung cells or your kidney cells. So bacteria that have capsules are generally nasty types of bacteria, and this is what a capsule is. So Streptococcus pneumoniae bacteria that cause pneumococcal pneumonia, which is a very bad lung infection, are bacteria that have capsules. So if you have a patient come to you and you suspect that they have pneumonia, You would send it off to lab, and in lab they would do a special stain, and if they see a capsule, we talked about the capsule stain, they may determine that you have a Streptococcus pneumoniae infection, and they would have to give you very specific types of antibiotics. So that's what a capsule is. And also it prevents the bacterial cells from phagocytosis. Phagocytosis is what your immune system does. So your immune system does something called phagocytosis, which means it eats up cells. So bacteria that have a capsule are not phagocytosed, so meaning your immune system doesn't really target them. That's why they're nasty infections to have. So that's the function of the capsule, to have cells stick to surfaces or each other, to protect them, and to prevent phagocytosis. Another structure that we're going to talk about in prokaryotes is flagella. Some prokaryotes, some bacteria have flagella, some don't. The function of flagella is motility. So let's talk about the structure. So just the main thing I want you to know is the function of flagella is motility. That's all I want you to get. So these are pictures of different bacteria. And so here, if you have flagella all around the cell, we call that peritrichous. If you have it on one side, it's polar, monotrichous is one. And then if you have lophotrichous, it's a lot. Polar is on one side, and then amphitrichous on each side. But the function of flagella is motility. So when we look at flagella, it helps bacteria move, and it's found outside the cell, so it's external to the cell membrane, and it is made up of a protein called flagellin. So in bacteria, if they do have flagella, it's made up of a protein called flagellin. And not all bacteria are motile, so not all bacteria have flagella, but if bacteria are motile, they have flagella. And here's a good video to explain it.
Cells with a single flagellum are monotrichous. When the flagellum rotates counterclockwise, the bacterium runs in a single direction. When the flagellum rotates clockwise, the bacterium tumbles and changes direction. Amphitrichous bacteria have a flagellum at either end of the cell. Bacteria with a tuft of flagella at one end are called lophotrichous cells. When the flagella rotate counterclockwise, the flagella bunch up and the cell runs. When the flagella rotate clockwise, they separate and the cell tumbles. Peritrichous bacteria have flagella covering the surface of the cell.
So all I want you to get from this slide are that flagella are important for motility, and you can have different patterns. When we see flagella all over the cell, we call it peritrichous. At just one is monotrichous, at each end is amphitrichous, and a lot at one end is lophotrichous. So that's the main thing. And then remember, flagella is made up of the flagellin protein. When we look at the structure of flagella from inside out, inside is the basal body. So the basal body is attached to the cell membrane, and then you have the hook, and then you have the filament on the outside. So when we look at this cell has a cell membrane and it has an outer membrane. So I know that this cell is gram-negative bacteria because it has an outer membrane. This cell structure does not have an outer membrane, so it's gram-positive bacteria. But whether you have flagella and you're gram-negative or gram-positive, you have the basal body, which is attached to the cell membrane, then the hook, then the filament. That's what I want you to get out of this slide. Okay, and now we're going to talk about Fimbriae and Pili, which are also important prokaryotic cell structures. So Fimbriae are these small, tiny hair-like structures that we see on some prokaryotic cells, and they're important for attachment. Not all bacteria have Fimbriae, but if they do, it helps them attach to surfaces and also helps them stick to each other. Pili, they're these also little hair-like appendages, and pili are important a little bit for motility. The main structure for motility is flagella, but where pili are important is for conjugation. I'm going to teach you what this word means later on, but bacteria can do this thing where they can exchange genetic material between each other. It's a really cool process where if you have, like, for example, one bacteria that is one color and one bacteria that's another color, they can touch each other through their pili if they both have pili and do conjugation, what we call conjugation. And that means just exchange genetic material. So transfer DNA from one bacterium to another. So if I ask you what is the function of pili, I want everyone to say conjugation. And we'll learn about it later on. Bacteria can have one or two pili, and not all bacteria have pili. But if you do conjugation as bacteria, you have to have pili. So again, Fimbriae are for attachment, pili are for conjugation, and brief motility. The main structure for motility is flagella. And then cytoplasm is a substance that we see inside the plasma membrane. So just inside the cell is the cytoplasm. It is just everything inside the cell membrane is a cytoplasm. It consists of the watery component that's called the cytosol and all the internal structures such as ribosomes and other things. And then there's also the cytoskeleton, which are just proteins that hold everything together. But I want you to know that the cytoplasm is just all the substance inside the plasma membrane. The cytosol is just basically the water inside. Inside prokaryotic cells, we see a nucleoid, which is not a nucleus because there is no nucleus in prokaryotic cells, and we're talking about prokaryotic cells now. Nucleoid is just the region that contains the DNA. So if you're looking at a prokaryotic cell under a very fancy microscope and you see the region of DNA, you call that the nucleoid. And in prokaryotic cells, the DNA is found as a circular double-stranded chromosome. Chromosome and DNA is the same thing. It's supercoiled, meaning it's tightly squeezed on each other. So that region where DNA is found on prokaryotic cells called the nucleoid. Some bacteria also have internal structures called plasmids. Plasmids are just extra pieces of DNA. We'll learn more about them later, but for now I want you to just know that it's just extra pieces of DNA. So they're extra chromosomal DNA, and it's extra DNA. It's not the original chromosome. So it carries non-crucial information, so non-crucial genes. Examples of genes and DNA are the same thing. Examples of genes that we could see on a plasmid that bacteria can have are antibiotic resistance. So it's an extra piece of DNA. Examples are maybe some plasmids give bacteria the ability to break down like milk or something, so nutrient use, toxin production, just extra non-crucial genes, but they can make a bacteria really good at causing infection. So bacteria can exchange plasmids with each other or DNA through their pili if they have it. They can do conjugation. Bacteria can have no pili, they can have one, they can have hundreds. Not pili, plasmids. So they can have no plasmids, they can have one plasmid, they can have many different plasmids. So keep that in mind. And again, to gain a plasmid or extra DNA, you have to have pili, and you do it through the process called conjugation. And you can gain or lose plasmids over time. Another internal cell structure that we see in prokaryotes are ribosomes. Ribosomes, their function is to make protein. So I want everyone to know throughout the whole semester, if I ask anyone, what is the function of a ribosome, is to make proteins. So we see ribosomes in all types of cells, prokaryotes and eukaryotes. The structure of a prokaryotic ribosome is it's made up of two subunits called the large subunit and the small subunit. We call the large subunit the 50th subunit and the small subunit the 30th subunit. The whole ribosome in prokaryotes is called the 70th subunit. Now, I know 50 and 30 do not equal to 70, but they're not supposed to equal each other. The S stands for sediment, so meaning if you put a ribosome in a machine and you spin it really hard, it's how long it takes it to settle. That's where this 30S and 50S comes from, just if you're wondering where these terms come from. But what I want you to take from this is that ribosomes in prokaryotes are called 70S ribosomes, and they're made up within the whole 70S ribosome. It's made up of a 50S subunit and a 30S subunit. And so again, prokaryotes have 70S ribosome. Eukaryotes, we have completely different ribosomes, which we're going to talk about later, but I want everyone to keep this number in mind, 70S ribosome. Prokaryotes can also, bacteria can also have these structures called endospores, or they can make them. Endospores are so bacteria sometimes encounter very stressful environments. Stressful meaning there's lack of nutrients, there's heat, you're putting chemicals to try to kill them. So some bacteria can form these structures called endospores. And what endospores are these dormant or resting forms of cells to resist poor conditions. So when there's really, really poor conditions and bacteria do not want to die, some bacteria, not all, have the ability to form endospores. So there's structures that are dormant that are made to resist poor conditions. For example, bacteria that do make endospores can make them when there's no nutrients. They're resting cells, they're not reproducing, they're just resting until conditions are good again. And endospores are very hard to treat. So if you are like the Clorox company or you're a pharmaceutical company, they always try to come up with drugs or cleaning supplies that can target endospores because they're very, very hard to treat. They're resistant to heat, drying, chemicals, radiation, so many things. So bacteria that form endospores can be very pathogenic or can lead to a lot of different diseases. They can survive thousands of years. And examples of bacteria that can make endospores are Bacillus and Clostridium bacteria and many others, but these are just examples. We'll talk more in detail about for example, the diseases that Clostridium can cause. Clostridium can cause botulism. And then there's a lot of contamination concerns. So this is why surgical instruments are sterilized. So they're put under a lot of cleaning procedures and sterilization so that we can get rid of endospores. So they resist poor conditions. And when cells are in unfavorable conditions or poor conditions, when they form endospores, we call that sporulation or making a spore or endospore. When the environment is good again, cells want to reproduce and go back to their natural self so they can germinate or return to their vegetative state. So germination is when the endospore goes back to its vegetative state because the conditions are good again. And I want everyone to know that forming an endospore is not a form of reproduction. It's just that environment is bad, so the cell forms an endospore, and that's sporulation. And then if the environment is good again, it'll germinate. And then we're going to talk about bacterial cell shape or morphology. So when we look at the cell shape of bacteria, we call that morphology in science. I just want to add a few slides here that I think are important for you before we end this lecture. So the average size of bacteria is about 1 micron, and we talked about it with the microscope lecture, so I'm going to skip the size, but I'm going to go to the shapes of bacteria. The morphology of bacteria have characteristic shapes, which we call morphology, and it's important to know the shape of bacteria for clinical and research aspects. So maybe if you know that your bacteria that's causing someone's throat infection is round and it's gram positive, you can say it's Streptococcus bacteria. So it helps scientists and doctors and pharmacists better determine their treatment option when they know the morphology. So it's important in bacterial species diagnosis. The different shapes or morphology that we see for bacteria are bacillus or rod-shaped. So bacteria that are rod-shaped, we just call rod, but all old textbooks used to call them bacillus, but just know that some bacteria look like rods and they're also called rods. Some bacteria are circles, we call them coccus. It would be so convenient if they were just called circles, but they're called coccus. So bacteria that are circular are called coccus. Bacteria that are rod-shaped are just called rod-shaped. And then some bacteria are spiral in shape. So these are the 3 different morphologies. So bacteria can be rod-shaped, they can be circular, they can be spiral. There are other unique ones, but these are the main ones. So most bacteria are either rod-shaped, circular, or spiral. And this helps us determine what the bacterial species diagnosis when you look at it under the microscope. So when we see pairs of bacteria, we say diplo. So when I tell you bacteria are diplococcus, it means that 2 circles are together. Some bacteria, that's the morphology of them. There are 2 coccuses that come together. Diplobacilli are 2 rods. Chains or strepto, chains means strepto. So streptococci bacteria are just lots of circles, so lots of coccus. Streptobacilli bacteria are lots of rods and chains. So that's chains. So if you look at the microscope and you see chains of rods, I would call this streptobacilli. If I see chains of circles, I would call it streptococcus. And then clusters, just like a wall in a group, are called staphylum. So staphylococci are just clusters of circular bacteria or coccus bacteria. And when I show you microscope slides from the lab portion, this will make more sense. So if I told you, not how can you identify, what would streptococci bacteria look like with a microscope? Just from the name strepto, I know that they're chains, and are they rod or circles? It's coccus, so they're circles. So they're chains of circles. So this right here is what they would look like. This here is streptobacilli. So to go off this slide, I'm sorry I cut off earlier, here is a picture of a bacterium that was gram stained. And just by looking at this image, I know if it's gram positive or gram negative. So it's purple, so I know it's gram positive because it's purple. The morphology, is it rod, coccus, or spiral? I can see that they're rods. And by the way, circles are perfect circles, coccus. So anything that's not a perfect circle is a rod. So that's why this is important. And now just to recap everything, we talked about prokaryotic cell structure, we talked about the plasma membrane and its importance for protecting cells and transport. These are the functions of the plasma membrane. The structure, it's made up of a phospholipid bilayer with proteins all throughout. We talked about the cytoplasm, that's just everything inside the cell membrane. So that consists of everything inside plus the watery component, which is the cytosol. We talked about the nucleoid, which is just the region of DNA in prokaryotic cells. Ribosomes, there are 70S ribosomes in prokaryotes, and their function is to make protein. And then some bacteria may have a cell wall. So a cell wall may be present, maybe not. We talked about gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria have a thick peptidoglycan layer and they stain purple. Gram-negative bacteria have thin peptidoglycan and an outer membrane and they stain red. And then Mycoplasma bacteria have no cell wall. And Archaea, if they do have a cell wall, it's made up of pseudomorine, not peptidoglycan. Then we talked about the capsule. The capsule is like a gel-like material outside of the cell wall, and it leads to more protection. And we talked about flagella, which is important for motility. Pili, which are hair-like structures all around the cell for attachment. And I apologize, Pili, Fimbriae, which I should have added here, Fimbriae, which is for attachment. Pili are the hair-like structure that are important for conjugation, which is when bacteria can change DNA with each other. And then some bacteria may or may not be able to form endospores, which are dormant structures that help cells during unfavorable, stressful environments. So here are the study questions for you to look at, and just take time with this lecture. It's a lot of material, so just stop it when it seems like it's a lot, and I hope that everything made sense.